Okay, now let's talk about what happens after transcription. So in prokaryotes, not much happens after transcription. In fact, really, as transcription begins, or as soon as transcription begins, translation starts. So if we go back, if you can imagine a given RNA being formed in a bacterial cell, a ribosome will be waiting right here at the 5' end to just start making a protein. And that's because in prokaryotes, like bacteria, all of this is just happening in the cytoplasm. There's no nucleus, so the DNA, the RNA, RNA polymerase, ribosomes, everything is all just floating around in the same medium. But in eukaryotes, the process of transcription is happening entirely in the nucleus, whereas translation happens exclusively in the cytoplasm. So there's some time and some steps that occur in between the formation of the RNA and the beginning of translation. And so for most genes for eukaryotes, there are 3 extra steps that have to happen before the transcript is ready to be translated. And that is 5' end capping, splicing out of sequences called introns, and the addition at the 3' end of a polya tail. So adding the 5' cap usually just involves adding a modified base called 7-methylguanosine to the 5' end. This is carried out usually by a complex of proteins which are actually attached to the RNA polymerase. So this is, again, an example of an RNA polymerase in a eukaryote. And so one of the enzymes that's part of that complex will basically grab the 5' end as it's coming out and add a 7-methylguanosine to the 5' end. And the purpose of the 5' end is to protect the RNA from an RNA nuclease. So remember that a nuclease, like an exonuclease, in this case an RNA exonuclease, is an enzyme that would break down RNA. And there are nucleases in the nucleus and in the cytoplasm whose job it is to break down and degrade used up or old RNA molecules. In fact, they're also important for protecting from viruses. So as we'll talk about, some viruses use RNA as their genetic material, and both prokaryotes and eukaryotes have enzymes that are there in part to degrade those RNA virus particles. So the 5' cap basically protects the messenger RNA from the cell's own RNA degrading enzymes. It's also important in the translation process because the ribosome and the complex of proteins involved in initiation of translation can recognize that 5' end. The next step is splicing of introns. So an intron is a sequence of bases that is found within a gene, but that does not get translated into protein. So introns do not exist in prokaryote genes because in a prokaryote, the gene is transcribed and then directly translated. So every base in the gene is translated into a protein. But the eukaryote genes have these sequences that do not get translated into protein. The only parts of a gene that get translated are called the exons. So every gene is made up of 2 or more exons and 1 or more introns. And each intron has to be spliced out before translation begins. And so splicing basically just means cutting the ends of the intron out and rejoining the ends of the exons back together. And there are different intron splicing mechanisms. These introns are called group 1 introns, and they are basically self-splicing. So there are certain sequences at the intron-exon border that trigger the reaction, this reaction in which a guanosine actually reacts with this adenine and breaks it off, breaks the sugar phosphate backbone, and then a new phosphodiester bond is formed between this uracil and this uracil. And so you end up with all the bases in between getting spliced out, and then the two ends, those two uracils, getting spliced together. So some genes contain introns in this group that basically splice themselves. Actually, that's something else interesting about RNA, is that RNA, unlike DNA, has the ability to not only act as an enzyme, actually carry out some enzymatic reactions, but they have the ability to react with themselves. And then these are, so again, those are group 1 introns, these are group 2 introns. Same idea, just the exact reaction steps are slightly different. So the intermediate that forms is slightly different, involves a different sequence of bases. And then other introns are spliced out using enzymes. So there are specific enzymes that recognize the intron-exon border, bring together those ends, cut the introns at the ends, and then rejoin the exons together. An example of the proteins that are involved in this process, some of them are called small nuclear ribonuclear proteins, and they actually themselves have RNA molecules they carry around. So this is an example of another role of RNA that's not translation. So small nuclear ribonuclear proteins are proteins in the nucleus that also have small RNA molecules attached to them, and they're part of this process as well. Now you might wonder, why would a gene have sequences of bases in it that don't get translated into protein? That seems kind of dumb to carry around DNA that's just going to get thrown away, because that's what happens to most introns. They don't, after they're spliced out, they simply get degraded. And the answer to the question, probably, of why splicing exists is because a given gene can be spliced more than one way. So, for example, this is a gene that has at least 6 exons, so 6 regions that can be translated into protein, but they are spliced at least 2 different ways. This is called alternative splicing. So in the thyroid gland, so that's a hormonal gland or an endocrine gland in the body, the cells that express this gene splice only these 4 exons, 1, 2, 3, and 4, and then everything after that simply doesn't get expressed. And so you end up with this transcript, which has exons 1, 2, 3, and 4. All 4 of those get translated into a protein, and that protein, after it gets processed by a protease, is called calcitonin. In the brain, meanwhile, this same gene is spliced this way. So the whole thing is transcribed, but then this region, which over here we called exon 4, actually gets spliced out. So this region over here acts as an exon. Over here, it essentially acts as an intron. So what you're left with is exons 1, 2, 3, 5, and 6. And so a different sequence of bases makes up the mRNA. It also gets translated into protein and then cleaved by a different protease, and you're left with this protein called CGRP. So 2 different proteins with different amino acid sequences being transcribed or expressed from the same gene, just in different tissues. So that probably is why alternative splice, or why introns exist, is because of alternative splicing. So it allows you to express more than one protein from the same gene, and it makes the genome of eukaryotes essentially a little bit more efficient, because the total number of genes in the genome for let's say humans is around 20,000, and that may seem like a lot, but it's actually a lot smaller than the number of proteins, in fact smaller than the early estimates for how many genes the human genome had. So before the Human Genome Project, the estimates for the total number of genes was in the millions, but once the Human Genome Project was completed, it was clear that there were a lot fewer than genes than that, so around 20,000. And that's only possible because of alternative splicing, because many of those genes can be translated into different proteins. So that's probably one of the main reasons why introns exist. But again, they only exist in eukaryotes because, yes, in eukaryotes, because in prokaryotes, they wouldn't have a chance to get spliced out before translation starts. And then the last extra processing step that eukaryotes have to carry out is the addition of a poly-A tail. So the poly-A tail is added at the 3' end, and what happens is the RNA polymerase gets to the end of the gene. It passes a given sequence that, once transcribed, binds to a different set of proteins that's part of the polymerase complex called polyadenylation factors. When the polyadenylation factor recognizes this enzyme, an enzyme called polyadenylate polymerase binds to it and just starts adding A's. It just adds adenine residues by bringing in ATPs, and just starts adding them one after the other to the 3' end, and it may add 10 or 50 of these in a row. Now, of course, these don't get translated. They get cleaved off before translation starts, or even if they don't need to be cleaved off necessarily, because translation will stop at a what's called a stop codon, which is part of the gene. So their job, the job of the poly-A tail, is similar to that of the 5' cap. It helps delay degradation of mRNA. Again, an exonuclease that recognizes RNA will break down RNA. And at the 5' end, you have the 5' cap, which completely blocks those exonucleases that start at the 5' end. And there are also exonucleases that recognize RNA at the 3' end. So this poly-A tail doesn't stop that from happening. The poly-A tail does not prevent degradation. It just delays it, because if you have a poly-A tail, what you basically have is a sequence of bases at the end of the strand that you can use to protect the RNA. So if this is your RNA, and then let's say this is an RNA exonuclease, so this is an RNase that starts at the 3' end. Well, it's going to start here as soon as it can, and it's going to move this way. But if there is a poly-A tail, so a bunch of adenosines here at the 3' end, and our enzyme it just takes longer. So because it has to get through all these A's before it gets to the actual important part of the mRNA, it just slows down that process. So the job of the poly-A tail is not to prevent degradation, just to slow it down. Because you want the RNA to eventually be broken down. Genes are usually only expressed temporarily. So once a gene is expressed, eventually you want the expression to stop. And one of the ways to stop expression of a gene is to break down the mRNA. So mRNA molecules are always sort of temporary structures. That's an important difference actually between RNA and DNA. So that's actually the end of that chapter. There's a lot more in your book that it goes over, but I decided just to cover the basics. So we'll stop there, and next time we will talk about translation.